Good evening, children. In our last discussion, we discussed about the structure of tobacco mosaic virus with the diagram, the labelings. Then we talked about the bacteriophage structure, which is a bacterial virus with the shape, all the parts, labelings and structure and functions. Then we talked about some many relevant technical terms, some meanings which are not in your books, but I had given you various definitions and terms, right? So these we had discussed up till our last lecture. Today we are going to talk about viroids. Though your syllabus says that only the definition is there, but because you cannot just directly cram up, so we need to understand a little about viroids. So what are viroids, children? These are the smallest known self-replicating particles. So are they smaller than viruses? Yes, right? So if you remember, in our last class, I had asked you a question and given the answer also, that is there anything smaller than virus? And the answer was yes. And the two substances are prions and viroids. So if prions are only the proteins that can invade the cells and somehow direct their own duplication, then viroids were only the RNA, right? So we are talking of viroids today. That what are these? These are smallest known self-replicating particles, and in the year 1971 they were described by I. Dana, who was a plant pathologist. Pathologist means used to study the plant diseases. Then they are smaller than virus and composed of RNA only, single circular strand of RNA of low molecular weight. They are made of about 270 to 380 nucleotides. Now what are nucleotides, children? You'll have to just revise. In your class 10 syllabus, you are studying nucleotides in structure of chromosome. So nucleotide is an assembly of a pentose sugar. If you are studying DNA, then this sugar is deoxyribose with a phosphate group at one side and a nitrogenous base out of any four. So what are these four nitrogenous bases? ATGC, that is adenine, thymine, guanine or cytosine. At a time, any one would be present. So if this is the case, that is in DNA. Right? So nucleotide is three things once again: pentose sugar, which is deoxyribose, phosphate group and a nitrogenous base out of any four. But when you talk about the nucleotide in RNA, children, everything remains the same except the nitrogenous base thymine is replaced by uracil. Spelling is URACIL. So this will be replacing thymine of DNA. The rest of the things, they remain the same sugar, but yes, one more difference. In sugar, if it's here deoxyribose, then here it is ribose sugar, right? Pentose sugar, that is ribose. Phosphate group would be as it is, and nitrogenous base becomes AUGC, that is adenine, uracil, guanine and cytosine. So that's the difference when you talk about the nucleotide of a DNA or a nucleotide of RNA. Coming back to our topic. So just now I told you that viroids, they have around 270 to 380 nucleotides. Then they are not surrounded by a protein coat. This is what you saw previous classes in the viruses, that they have a protein coat. Like in this TMV, the RNA helix was there and it had a protein coat called capsid, or that is made up of subunits called capsomere, right? But here that protein coat is missing. Alright, then they appear in nucleus of plant cells. So that is these viroids, they appear in the nucleus of plant cells and they cannot produce their own proteins. Why? Because of absence of initiation complex. This is we can say a very important step for the protein synthesis, and because this complex is absent in viroids, they cannot make their own proteins. What diseases they cause? They cause diseases like PST, that is potato spindle tuber disease, right? Chrysanthemum stunt disease. Stunt means to remain dwarf, that is the normal growth cannot take place. Then cucumber pale fruit disease and tomato apical stunt disease, right? They enter the plant, the crop plants, from some unknown wild plants, and in fruit trees they spread through grafting. All right, so grafting, as we know children, one of the vegetative propagation steps. This is spread through grafting in the various fruit trees. That's all about viroids children, right? So with that topic of viroids, we come to an end of the unit on virus. I'll just show you what all syllabus you need to do. I have already sent you virus from the beginning till the end. This is the only thing that you need to study. Characteristic features, link between living and non-living, that we have done, why it behaves like living or non-living, structure of TMV, we have done it, and bacteriophage, and the contribution of various scientists like D.J. Iwanovsky, M.J. Bijring, W.M. Stanley, and viroid definition only. So no need to go much deep beyond that, but still we have discussed here, apart from definition, like who discovered and what all things it's missing with, like it's missing the initiation complex, therefore it can't make its own protein, and what all diseases are the ones that are caused, and that's all about viroids. So with this topic children, quickly let us revise what all things we have done from the beginning, because today we are ending up viruses and we are going to start our new lesson about body fluids and circulation. You should have compiled up all these notes at one place children. Keep revising from time to time, because at the same time you're also revising for your remaining exams for ICSE, but tabulated view of these will make your study easier and systematic. I'm just showing you what all things have I sent you, right? That will complete your topic over here. With this, some technical terms, right? And today's viroids, right? Prions, though I have made notes, but I am not giving because that's not required to be in your syllabus. But just one thing you should know children about prions, that they are only proteins that can invade the cells and somehow direct their own duplication, right? So they are infectious protein particles without nucleic acid, right? And the details though I have written, but not necessary because it's not given in the current syllabus, so we will not do this. Alright, so I'm sending you only the relevant notes, please make up. And today I'll also start with your new topic on circulatory system. Thank you.